This program is presented by University of California Television. Like what you learn? Visit our website or follow us on Facebook and Twitter to keep up with the latest UCTV programs.
So these are the Berkeley graduate lectures, the Hitchcock lectures. And as you heard yesterday when Carol gave her first talk on how telomeres cause age-related disease, there's a great story in discovery that can be serendipitous. And so today, Carol, I'll introduce her shortly, will give us a story that's been retitled The Anatomy of a Scientific Discovery. She's going to tell you really much more about the process of how scientists discover things that are later deemed to have high impact, as we all would agree, from the 2009 Nobel Prize in Physiology or Medicine. So Carol is a professor and director of molecular biology and genetics at Johns Hopkins, but she started as a California native. She was born and raised here for the most part, and she went to UC Santa Barbara for her undergraduate degree, and then came here to UC Berkeley for her PhD. And from there went to Cold Spring Harbor Laboratory as faculty, and in '97 then to Johns Hopkins. But it's from Cold Spring Harbor, from the early '90s, that I know Carol, where I had the pleasure of joining her research group. And there are many things that you'll hear that are really important features in a scientist who's committed to doing discovery, basic research. And Carol exemplifies, really, those features. And I think, in part, the MCB graduate program here and Carol, to me, have the important starting point, as she'll talk about, of curiosity-driven research and hard work. But I say that it takes much more than just curiosity and hard work, because it also takes the patience to let the question unfold, to let the real question that you should be answering unfold, as opposed to the question you started asking. And also it takes the insight to recognize when the question you should be asking comes along. And I'll just quote, in fact, from the 1945 winner of the Nobel Prize in Physiology or Medicine, Sir Alexander Fleming, whose comment was, "One sometimes finds what one is not looking for," in describing his discovery of penicillin, which he discovered accidentally. He's a bacteriologist. He loved to watch and understand how bacteria grew. And he had stacks and stacks of plates from his curiosity and his hard work, and he put them aside. And one day, he happened to notice in the corner of one of these plates a giant colony of mold. And you know, this is not what he set out to do, to grow mold. But he noticed something about this mold, and it was this insight that led him to penicillin. He noticed that this particular mold wasn't just competing with the bacteria for space on the plate. It was actually killing the bacteria that surrounded it on the plate. And from there, he recognized, he had that insight that, this could be a new question to study, as opposed to the question that I started this experiment with in the first place. And I've learned a lot from Carol, and I've learned a lot from this process of learning not just to be curious, but to have more patience and to let the questions change and evolve and take you to what you weren't sure you were asking in the first place. So thanks, Carol, and welcome back to Berkeley.
Well, thank you very much for that perfect introduction for what I'm hoping to tell you today. So yesterday we talked a little bit about the science of telomeres and telomerase. And today I thought that I would take off a little bit and use telomerase and the discovery of telomeres as a way of telling a story about exactly that, about how serendipity and chance play a role in the progress of science. So once again, as we were discussing yesterday, we'll be discussing telomeres. So for those of you that weren't here yesterday, I'll just go back over this. Within the nucleus of a cell, you have chromosomes, and the genes are all along the length of the chromosomes, and the telomeres are these end parts that protect the ends and maintain the length. And so we'll be talking a little bit about some of the discoveries along the path to understanding the role of telomeres in a number of different organisms. So if you think about scientific discovery, in hindsight, it may seem that the discovery is a fairly linear process. So telomeres were first recognized as important components in the 1940s. And then the telomere sequence is identified. And then it was clear that there was a role of telomere elongation in telomere length maintenance. Telomerase was predicted. Telomerase was then discovered. We then understood the connection of telomeres and cancer. And then telomeres in disease, and then in 2009, a Nobel Prize for the discovery of telomerase. So it may seem like this is a linear process, when in fact, the process is really much more complicated than that. So what I'll be doing is explaining how, in reality, the process is very messy, involving serendipity, personal interactions, insight, hindsight, and various other approaches. So I don't expect you to understand all of this yet. We'll kind of go through it and work our way through. But there are 2 points that I want to make at the beginning. The first of all is that this timeline is not an accurate linear timeline. You'll see that the 1980s are greatly overly represented here. There was a lot of particular progress that went on in the 80s and the early 90s. So this isn't meant to be an accurate linear representation of time. But also, I'm taking a fair amount of artistic license here. So this is not meant to be any kind of a definitive history, and I apologize in advance for anyone whose important discovery I might not mention. This is really meant to be more metaphorical about how the process of discovery unfolds. So this isn't meant to be taken as a definitive history by any means. But there are going to be some themes that I'm going to bring out during this talk. And the first is that curiosity is a driver of science. Discoveries tend to happen in batches. That's in part because people talk to each other. But it's important that discoveries happen in batches because replication is really essential to establish a new discovery as something fundamentally new. Fields collide and inform each other. So you may be working in one area and then start working in another area, and it's the juxtaposition of those fields where a lot of new things can usually be found. And scientific discovery is not like engineering. Changing direction or objectives can be a good thing, very much like what you just heard from Kathy, being able to be distracted by something that may be the question that you really should ask. And then finally, another quote, this one from Louis Pasteur, that chance favors the prepared mind. So we'll be working through some of these themes as we go through, as an example of a scientific discovery, some of the discoveries about telomerase. So the telomeres were first functionally defined as units that protect chromosome ends in the late 1930s, early 1940s by H.J. Muller working in Drosophila and Barbara McClintock working in maize, showing that there were functional units that had to protect chromosome ends. But then that initial recognition of these functional units had to wait a number of years before we could really understand anything about the mechanism of what these functional units actually do. So it was in 1972 that there was a paper that was published by Jim Watson, one of the co-discoverers of the structure of DNA. And what he defined is what's called as the end replication problem. The problem with replicating the end of any kind of a linear DNA molecule. And so if you have such a linear DNA molecule, it turns out that what was known in the 1970s was the mechanism by which this replication occurs is that you start copying this chromosome from an internal location. And when you then copy to the very end of the linear molecule, it turns out that there's a small region, because of the biochemical mechanism by which these are made, there's a small region at the very end of the chromosome which isn't copied. And as you go through this process and continually copy many chromosomes, you then expect that chromosomes would shorten progressively from their ends. So this was a theoretical paper that was published by Jim Watson. And around the same time, it turns out that there was a paper, also a theoretical paper, published by Alexei Alovnikov in the Journal of Theoretical Biology. Although he did not use the term telomere in this paper, he noticed that there may be a problem with copying the ends of molecules, and he used this term marginotomy, being the margins of the chromosomes, instead of using the word telomere. And because of that different usage of words, his publications weren't well known to the telomere community until a number of years later. However, Alovnikov did show that, that there was this theoretical problem of copying the ends of chromosomes, and he had the important insight to link that to a paper that had been published in the 1960s by Len Hayflick, which suggested that if you take primary human cells and put them in culture, that they have a limited lifespan in culture. So this limited lifespan of human cells was termed cellular senescence. So back in the 70s, these two ideas were linked, but as you'll see, we weren't able to follow up on what that meant until a number of years later. So the first molecular characterization of telomeres, and this is one of the themes that we'll bring out, is that as soon as you identify actual components and the molecular mechanisms, it's easier to understand function. So the first molecular characterization came from the identification of the telomeric DNA sequence in Tetrahymena, this single-celled organism that has 40,000 chromosomes. And what's shown here is a nice electron micrograph of this particular molecule, which is important from Tetrahymena, which was a 21 kilobase molecule that has telomeres at both ends. And so Elizabeth Blackburn and Joe Gall were very curious about what was the actual DNA sequence that was at the ends of those chromosomes. And they published this paper in 1978. And what they showed is that there were many copies of this very simple sequence on both of the ends of these molecules. And so that then defined the telomere repeat sequence for the first time in any organism as these tandem repeats of simple sequences. So this paper was in 1978. Shortly thereafter, just 3 years thereafter, there was another paper, again from ciliated protozoa, in this case Oxytricha, which looks a little bit like its cousin Tetrahymena, although they are actually quite distantly related. And what this group showed was that there was a very similar tandemly repeated DNA sequence on the ends of the chromosomes in 4 other ciliates. So in this case, this Oxytricha telomere sequence was tandem repeats of the sequence T4, G4. So there's T4, G4, T4, G4. And then they showed at the very end, there's an extension on one of these DNA strands relative to the other. And it turns out that this was a very important functional characterization of the ends of the chromosomes. So the end replication problem should predict that telomeres shorten every time cells divide because you can't completely copy to the end of the chromosome. And so in understanding this as a theoretical problem, there was sort of a mindset as to, okay, how are we going to overcome this telomere shortening? However, telomere shortening was not seen when you were growing these Tetrahymena cells in culture. So why was it that telomeres aren't apparently shortening as cells are growing in culture. In fact, it was found very shortly thereafter that telomeres would actually get longer in some single-celled organisms as you grew them in culture. And this was very counterintuitive. So unexpectedly, there was this telomere elongation. And this was a paper from another single-celled organism, trypanosomes. And if you look on this southern blot here, there's a band that does not change size as these cells are growing for increasing numbers of divisions in culture. But then this telomeric band here, as you grew these cells, it actually got longer, which is the opposite of what one had been thinking about with this end replication problem. So this didn't make a lot of sense. However, when you do the same experiment in Tetrahymena, where the telomeres were first identified, again, it was seen that the telomeres actually got longer. So here are cells grown for increasing numbers of cell divisions, and this telomeric band, you can see increasing in length as these cells are grown continuously in culture. So this is another example of discoveries happening in batches. So we really believe that this is the case, that there is this telomere elongation, but yet we don't know why that is. So in the early years of the telomere research, we were really gathering pieces of the puzzle together. They really didn't fit, but by gathering up all these various different pieces, then trying to interpret what may be occurring. So the telomeres were shown to be functional units, and then the end replication problem was proposed, and that was linked to cellular senescence. The telomere sequence was also identified, which allowed an understanding of what's going on at the molecular level, and then one was able to see this telomere addition. So that was sort of the early stages of trying to understand what was going on with telomeres in cells. The next important functional characterization came from this experiment that was done by Liz Blackburn and Jack Shostak, where they were able to show that Tetrahymena telomeres would function as telomeres in yeast. So here is a circular plasmid, which was very familiar to people that were studying yeast. And when you take that circular plasmid and open it up and add Tetrahymena telomeres onto the ends of these chromosomes, the Tetrahymena telomeres functioned as telomeres in yeast. And as I'll be saying in a few minutes, they were actually elongated in the yeast. But this paper was published here in 1982. And in 83 and in 84, again, similar kinds of findings were also made independently by other groups. So here's one paper which has a kind of unusual title that doesn't really tell you what's happening in the paper. But what they did in this paper, also published in Nature, was they showed that there must be GT-rich sequences which are present on the ends of chromosomes in yeast. And they showed that there was addition of GT-rich sequences onto the Tetrahymena telomeres. Also, in 1984, there was a paper that showed that when you take telomeres in yeast, if you take now, in this case, oxytricha telomeres, and put them into yeast, that those oxytricha telomeres would function as telomeres in yeast, and that they were elongated. So again, here's an example of discoveries happening in batches and reinforcing one another. So this general concept that telomeres would work in a variety of different organisms was clearly very well established. And then later in 84, the Shostak and Blackburn lab went on and identified the actual sequence which was added on to these Tetrahymena telomeres in yeast. And from this paper, they proposed that there may be some kind of an activity, telomere transferase-like activity, that would extend this GT-rich strand of the yeast telomeres. In the exact same journal where this was published in Nature in 1984, the next paper in the journal was a paper by a group that was also proposing a way that you could elongate telomeres. How is it that telomeres are shown to be elongated, and why would you have these sequences added on to the Tetrahymena telomeres? And I'm not going to take you through this complicated model, but basically what this group proposed was that there was a recombination mechanism by which telomeres could be added, as opposed to de novo addition mechanism. So I'll just show you a cartoon here. A recombination mechanism, you can imagine that if you have a particularly long telomere and then a short telomere, is that you can actually copy this one strand from the short telomere can invade the longer strand, and you get net elongation of the short telomere, and then repair synthesis. But the main point here is that you start off with a long telomere and a short telomere, and you end up with two long telomeres via a mechanism that was a well-known mechanism in the DNA repair field. In contrast, what had been proposed for the de novo addition is simply adding onto this 3' end sequences in this direction, and then filling out and getting net elongation by this mechanism. So the same set of data led two different groups to propose two very different models. So understanding that telomere addition may be occurring, one group proposed a recombination-based model, and another group proposed that there would be de novo elongation. So it wasn't necessarily clear how this would come out in terms of the importance to telomere replication. And as you will see, we'll be talking a lot about de novo elongation, although later on, recombination also will turn out to be important in addition. So, so both of these pathways will turn out to be important. So when I came to Berkeley to work with Elizabeth Blackburn, I was then charged with trying to understand, is there any evidence for something which would add new telomeres onto the ends of chromosomes? So to test one of those two models, which was the de novo elongation model. And so what we did was to take, again, Tetrahymena, which has a source of many telomeres, and we were hoping would have a source of many of the components which were necessary for making telomeres, and to grind them up and put them into a test tube with the right sort of reagents. And the reagents would be something which would mimic natural telomere end, and the precursors for the telomeric sequence, which is necessary to be made. And what we found was that this enzyme, Telomerase, would extend the primer by adding sequences of the telomeric repeats. And that is what is shown here, the elongation of the telomeres. So this was just 1 year later, after the initial discovery that the telomeres were added onto the Tetrahymena telomeres in yeast. Then by asking the question, how is it that you can actually have an enzyme add something that is not templated onto the ends of chromosomes, we proposed that perhaps there's a essential nucleic acid involved, that is an RNA component. And we identified this small RNA, which contains the complement of the telomeric repeats as part of the RNA, and showed that the way that the telomeres are elongated is by copying this template within the telomerase. So those discoveries happened in a relatively short period of time. So after we understood about the telomere addition and the de novo elongation, this predicted the telomerase. Telomerase was discovered, and the RNA component was identified just a few years thereafter. So we're basically collecting up the different kinds of components of the machinery that's necessary for maintaining telomeres. So another question that we didn't really know very much about that was going on about the same time was trying to understand from the standpoint of the telomere what's actually bound to the telomere itself. The telomerase would be the enzyme which would make this longer, but what is normally here along the telomere. And so a number of groups were interested in identifying what the components might be that would make up the telomere itself. And in a series of papers that were happening in the middle 80s, again, the telomere binding proteins were first characterized by Dan Gottschling and Tom Check, again, in ciliated protozoa, these organisms that have many tens of thousands of telomeres. Again, going to the source of where telomeres are most plentiful. So they found that there was evidence for a specific telomere complex. And then Dan Gottschling and Ginger Zakian were able to identify the specific proteins that were bound in that complex, and then finally actually cloning to know the individual subunits of this telomeric complex occurred in the early 90s. So the discovery of telomerase then was around the same time as the discovery of these telomere binding proteins, identifying the individual components. So at this point, what we knew about was that there were these telomere binding proteins, and most of the work had been done in either yeasts or ciliates, either Tetrahymena or Oxytricha. And it still wasn't necessarily clear to the wider community of biologists that what we knew about telomeres in these single-celled organisms really would be informative of anything that might happen in humans or in other mammals. That is, what relevance did this actually have to human disease? Now, certainly from the standpoint of a biologist, we knew that something that was very fundamental for all cells very likely would be conserved throughout many different organisms, although it wasn't necessarily clear to the scientific community at that time. So the first hint of what was actually going on in human cells came in the middle 80s from this paper that was published by Howard Cook's lab. And what they found was that there was a region on the X and the Y chromosomes which was very close to the human telomere. So this is just a cartoon of the human X and Y chromosomes. And of course, most of these chromosomes, most of the DNA on these chromosomes is different from each other. But it turns out that there's a region which is called a pseudoautosomal region, which has sequences that are present on both the X and the Y chromosomes. And Howard Cook was trying to understand this pseudoautosomal region. And what he came across was that when he probed for these probes, that the bands that he saw were very heterogeneous. And this looks very much like this heterogeneous sized band that was seen for telomeres in both Tetrahymena and in yeast cells. So here was a hint that the evidence that we had for what telomeres would look like in these single-cell eukaryotes may actually play a role in human telomeres. And they went on then to do really the definitive experiment at the time to show something really is telomeric. What you do is you take and you treat with a nuclease, and this is an enzyme which will chew away from the end. And this heterogeneous band here, then, if you chew away with it with a nuclease for increasing amounts of time, it decreases in size. So this suggests that it really is the natural end of the chromosome. So this was in 1985, and we didn't yet know the sequence of the actual telomere. Okay, so these bands, they are identifying because they're looking with sequences that are very near the telomere. So they still didn't know what the actual telomeric sequence was of the human sequence. It wasn't until 1988 when the Moises group was interested in looking at repetitive DNA sequences from humans. And they came across this repetitive sequence, T2AG3, and they were able to show that that repetitive sequence, when you hybridize that to human chromosomes, and you may be able to see here on the ends these faint yellow dots on these red human chromosomes, they were able to show that this repeated sequence is actually present not only on the human chromosomes, but also on many other vertebrate chromosomes, mouse, and a variety of other organisms. So this now definitively identified the human telomeric sequence, and this was really an enabling discovery because it allowed testing of models for what's happening with human telomeres. Again, the discovery in one lab leads to rapid discoveries in other labs. So the human telomeres and the entire sequence of the human telomere was then cloned by two different groups, again in back-to-back papers in Nature. And these groups used the exact same approach as had been identified for cloning the yeast telomere by Blackburn and Shostak, that is to take and put human DNA into yeast cells and showed that human DNA actually functions as a telomere. And they used nuclease sensitivity to show that cloned human DNA actually was telomeric. And this is once again another example of discoveries happening in batches. And this is important, not just because a lot of things are happening around the same time, but because by these very similar findings coming from different groups from different angles, it really established in a very short period of time the importance of these sequences. So we had been focusing on telomerase and the telomerase components, the telomere binding proteins, and then with the identification of the human telomere sequence, that then allowed us and others to go on and test models that had been around for a number of years. And specifically, the model that we were able to test was this question about telomere shortening in primary human cells as they divide in culture. As I said, it had been proposed in the 1970s that the progressive telomere shortening in human cells may play a role in cellular senescence. And so I was fortunate to collaborate to be able to ask this question. Very shortly after the identification of the human telomere sequence, we were able to show that if you actually take human cells and put them in culture, as Hayflick had done, and show that these cells divide for a certain number of times in culture and then stop dividing, and we were able to use the human telomeric DNA probe and show that indeed the telomeres shorten progressively as these cells are dividing in culture. Not only does this happen with cells that are dividing in culture in humans, but it also actually happens in human cells with age. So this is an example of the average telomere length taken from a gel like I just showed you, compared to the age of the individual, and that there's progressive telomere shortening with age in these human white blood cells. And now, having recently discovered telomerase and understanding that telomerase is necessary to add sequences onto the ends of chromosomes, what we initially thought was, well, these cells perhaps don't have telomerase, and so they're shortening progressively in the absence of telomerase. So that's what we thought then, that there was no telomerase in these somatic cells. But now what we think is happening is that although there may be telomerase activity in those cells, the telomerase activity is limiting, and the cell division can outpace the elongation that's occurring by telomerase. So within the lifetime of these individuals, there's so much cell division going on in the blood cell compartment that the division gets ahead of the ability of telomerase to elongate those telomeres, and that's why you see this telomere shortening. So as I mentioned, it was proposed a while before that cellular senescence may have that telomere length may have some role with cellular senescence. And so this is really the point that ideas aren't always connected in a linear fashion. That past work that we didn't really understand maybe how it fit in to the story can then become relevant later on. And in this case, it became relevant because we knew the human telomere sequence. And we needed to know what that sequence was in order to do the experiment to ask about the telomere shortening. So indeed, knowing this human telomere sequence then led us to understand about the cellular senescence, even though this had previously been published a number of years before. So along with understanding the telomeres and cellular senescence, the other thing that was quite enabling for understanding with the human telomere sequence was understanding about telomeres and cancer. The same year that we were looking at cellular senescence, Titze de Lange and with Harold Varmus were looking in both cancer cells and at different tissues. And what's shown here is the fact that there is tissue-specific differences in telomere length within people. So they're looking at telomeres that are quite long in one person, comparing the sperm telomere length to the blood telomere length. So these are two different tissues and two very different telomere lengths. Another example here, sperm versus blood, much longer telomeres in this setting. And then over here, what they found was that if you compare normal tissue to tumor tissue, that there was significantly shorter telomeres in the tumors. Again, normal and tumor, so there were shorter telomeres in tumor cells than in normal cells. And we now can understand this because that tumor cell has gone through more rounds of cell division than it otherwise would have due to some genetic change that allows it to divide, and that more rounds of cell division then allows this telomere shortening. So, as I had mentioned before, discoveries happen in batches. That very same year, there was another paper which was also published, showing in this case the normal is shown, normal telomeres here, and tumor telomeres from the same sample, normal and tumor, normal and tumor. So there's telomere shortening in cancer cells. So, discoveries happen in batches, and independent replication is very important for verification of fundamental ideas. So in addition to understanding the telomere length in tumor cells, it was interesting to know what is the role of telomerase activity in tumor cells. And so we were interested in the role of telomerase and when telomerase may be present in tumor cells. And so this particular paper, telomere shortening associated with chromosome instability is arrested in immortal cells which express telomerase activity, was the first example of telomerase being activated in cancer cells. But I do want to have a little disclaimer here that even though I'm an author on this paper, I wanted to just apologize, because one of the things when I was doing my PhD thesis here at Berkeley was that my colleague Jasper Ryan told me many times that you should not write cells which express, but cells that express. So I just want to take that as a disclaimer, and I will never make that mistake again. So this was a paper that showed that telomerase becomes activated in cancer cells, and then 2 years later, a definitive paper also showing that in most human cancer cells, you have telomerase activity in order for those cells to go on and continue dividing. So this is getting back to some of the issues that I raised yesterday in the talk, and that telomerase is required for cells that divide many times. So the fact that you see activation of telomerase in cancer cells, this one particular cell undergoes a genetic change that has to divide many times, and this cancer cell has to solve the telomere problem. And one of the ways it does that is by activating telomerase. After fundamental discoveries are made in a particular field, it turns out that there's can be a lot of activity in the field. And so what I've shown you so far is many of the papers that throughout the 80s, which were leading up to establishing all of the different components, the telomere binding proteins, the telomere sequence, the telomerase activity, to understand what might be going on with telomeres. And so if you look at the total number of publications with the word telomerase in it, across here, there were a number of publications, but it looks like it's 0. And then at this point, once we have enough enabling discoveries, the number of publications really just skyrocketed, and people started investigating more about different kinds of cancer cells and how much telomerase is there and what's going on with the telomere length. So this is really a common phenomenon in many fields once you reach a critical mass of understanding of the individual components. So we had understood the telomere sequence led to understanding senescence, and it also led to understanding the role of telomeres in cancer. It was very important, though, to understand the different components if we were going to understand the role of telomeres in human disease, to understand those components that are on the ends of chromosomes. And the first identification of the proteins that bind to the ends of chromosomes in human cells came from these two papers from the De Lange lab, identifying this protein TRF1 for telomere repeat binding factor and TRF2. And you can show in this nice set of chromosomes over here, now we're looking at the actual proteins that are bound to the chromosome ends, that these proteins are binding to the very ends of these human telomeres. And so the identification of TRF1 and TRF2 then led to the identification of what we now call this sheltering complex. So TRF1 binds along the length of the telomeric DNA, as does TRF2. So these were the first two components that were identified, and we now know of this complex of proteins that's needed to protect the human chromosome ends, and also plays a role in elongation of the telomeres with telomerase. So we still, at that point, although we knew many of the different components, we didn't know what the makeup of telomerase was. We knew it had an RNA component that provided the template, but the catalytic component of telomerase remained elusive. And it was, again, the ciliated protozoa that came to the rescue. So Joachim Ligner in Tom Check's lab first identified the catalytic component of telomerase from another ciliate called euploides. And then working together with Vicky Lundblatt showed that there was a very similar kind of an enzyme in yeast. And this is just one figure from their paper where they were able to show that the important catalytic regions of this enzyme that they had identified actually was similar to a reverse transcriptase. So this is an example of the HIV-RT, suggesting that this telomerase reverse transcriptase was the catalytic component of telomerase. And this was quite an important discovery, because now that the field had their hands on an actual molecule, you can then go and do experiments to test hypotheses. And one of the first experiments that was done after the identification of the TERT enzyme and knowing what the human TERT was to express that component inside human cells. And as I had shown you that if you look at cells that have been grown for a long time in culture, you have relatively short telomeres, as shown in these 4 lanes here. But as soon as you put back the telomerase, and so you add more telomerase to cells, what you find is that the telomeres are elongated. And in this case, this would increase the number of cell divisions that those cells would go through in culture, showing that the telomeres actually do limit the number of times cells can grow in culture. And this was quite an enabling discovery, because now in a number of different laboratories working in a variety of different cell types, people could grow cells and be able to study them for a longer period of time in culture. And so today, many different human primary cells can be grown for a long term in culture. If you simply put this telomerase back into the cells, you can now grow those cells because they're no longer limited by the short telomeres. So in terms of practical applications for scientists, this makes it easier to understand many different cell types. So the identification of the human telomeric sequence directly enabled the understanding of shelterin, and telomerase allowed the discovery of the TERT component, which then allowed many additional discoveries to be made. So the final question that I'll just leave you with here, I won't go through many of the other discoveries, but the question about what actually happens without telomerase. And this was the subject of my talk yesterday, but this is just an example of if you have mice that lack telomerase in the first generation, you cross those mice, you get second generation mice that have shorter telomeres. You breed those second generation mice, you get third generation mice with shorter telomeres, et cetera, for increasing number of generations, that this progressive telomere shortening in mice actually leads to a number of different complications. And what happens at the cellular level is that with each generation, the telomeres get shorter, and those short telomeres then trigger either cell death or cell senescence. And this was shown by a number of different groups. However, it turns out that some cells can actually escape this cellular senescence. Although most of the time what happens is the cells undergo cellular senescence, the short telomeres also play another role. And this was also something that was shown a number of years before, and that is that they can sometimes escape and they can undergo a recombination pathway that allows them to escape senescence. And so this is just a figure from this paper of Vicki Lundblatt and Liz Blackburn indicating that there is a bypass pathway that if you don't have elongation by telomerase, the telomeres can be elongated by recombination. And this is harking back to what was proposed about 10 years earlier. And that is the recombination at telomeres. So the discovery of telomerase led to being able to ask the question about what are the consequences of short telomeres. And remarkably, recombination is one of those consequences. And this is something that, again, the process doesn't necessarily work linearly. Recombination was proposed a number of years before. The cellular senescence is one of the consequences of telomeres. Initiation of the DNA damage response and cell death was another consequence. So the overall synthesis of this is that defining the molecular components together with understanding the consequences of short telomeres allowed the discovery of a human disease mechanism. So many of these components that we've been building up over the years then led to the understanding of the human genetic disease, dyskeratosis congenita. So understanding the telomerase RNA and understanding the consequences of short telomeres led to knowing that this disease is actually a disease of short telomeres, which then several years later made it clear that there are really a collection of different effects of short telomeres, often called the telomere syndrome. Understanding these consequences puts new understanding about telomere length regulation, and these experiments are really still the focus of many different laboratories today. So as I was concluding yesterday, discovery is not a linear process, and new discoveries sometimes come from unlikely places, Tetrahymena and Oxytricha. So some of the themes that we've been discussing here today: curiosity is a driver of discovery. Discoveries happen in batches. People talk to each other, and replication is essential to establish new discoveries. Fields collide and inform each other. Scientific discovery is not like engineering. You have to be ready to change direction and follow what's most interesting. And chance favors the prepared mind. And so with that, I'll just leave you with one final slide, and that is, where was telomerase actually discovered? Wendell M. Stanley Hall at UC Berkeley. Many of you may or may not recognize this. This is a nice vintage car sitting outside on the circle. And my final point today is that you can't go home again. This is what happened to Stanley Hall. So here's the remnants of how it used to look. And now there is a new beautiful building in its place. So I'll thank you for your attention.
Hi, thank you very much for your great talks. I read a biography last year about Henrietta Lacks, who was the donor for HeLa cells. Sorry, I'm a chemist, not a biologist, but it's my understanding that those cells were somehow unique in being able to avoid senescence and were used widely in molecular biology research and until recently. And I'm wondering if there's any understanding now of were those cells somehow able to use one of these mechanisms to avoid senescence?
Yes, so the HeLa cells are a cell type that is derived from cancer. So in the case of Henrietta Lacks, it was her tumors that were put into culture. And this happened for many other different cell types. And as I had shown, what happens is that telomeres do get short, but then telomerase becomes activated in cancer cells. So that's a widely common phenomenon. In the case of the HeLa cells, it happened that they were found to be very useful by people, and so they've been used in a number of different laboratories, as are other cell lines. So there isn't anything unique about those cells having activated telomerase, but they were a cell type that was very easy to grow in culture because they had been derived from a tumor, and so they were a very useful resource for understanding human cells, and so they were widely distributed to many researchers to understand human biology.
Thank you so much for your lecture. My question is, you said the study was done on rats, so the synergist effect of the cells getting short, you talked about that, but how do you make them grow? I mean, as far as diet and activity, how did you make the cells grow in the rats?
Do you mean the tumors or?
No, healthy, we want healthy cells, right, that are long?
So the experiment that I showed where you can take normal human cells, and normally the telomeres are shortening progressively, and then if you put back this enzyme telomerase, you can make the telomeres be elongated, and those cells will now grow for many, many more divisions because you no longer have the short telomeres that limit the growth of those cells. So those are normal cells that now have what we would call an extended lifespan or have become immortal.
Immortal. Okay.
I have two interrelated questions. Number one, in the cancer cell, you mentioned telomere length is shortened. So why does cancer cell not enter into senescence? Why does it keep on dividing? And second thing you mentioned in the cancer cell, telomerase activity is very high. So why does telomerase does not make the telomere length go back to the normal length?
So you hit on something exactly that's kind of counterintuitive. I did tell you that cancer cells tend to have shorter telomeres, and I showed you some evidence from some historical papers. Cancer cells have shorter telomeres, but cancer cells also have telomerase activity. And so it's that telomerase activity that maintains the telomere length in the cancer cells. So that's why they don't undergo senescence. So they started off with long telomeres. They went through a number of rounds of cell divisions where the telomeres were shortening, and then they got telomerase activity by, we're not sure exactly what mechanism, perhaps a mutation. Now they have telomerase activity, and now they're maintaining the telomere length. Why they don't go back up to the original level, we don't know the answer to that. That's a very good question. However, we do know that these are cancer cells, and so they don't have many of the normal regulatory mechanisms that normal cells would have. So we have actually done the experiment of shortening the telomeres in a completely normal organism, in mice. And then when you put telomerase back, you do end up going back to that normal set point because it's the normal regulation of those enzymes. But it doesn't often happen in the cancer cells, and that's because those are probably disrupted cells. But you're right, it's counterintuitive.
Sorry, I have one more question. What do you think if a cell has entered in a senescence, that cell can be reverted back to the active state, or this cell will remain forever for the senescence?
That is a good question. Once an individual cell has entered senescence, it is thought that you cannot drive it out of senescence. And these are experiments that were done before telomerase. If you can put in agents which will cause the cell to undergo DNA replication. In the experiments that I'm showing you where you take a culture of cells and all of those cells are heading towards senescence, now you put telomerase into the cells. Some of the cells are still actively dividing. They haven't yet gotten to that final senescence point. And so when you put the telomerase in, now you rescue those cells that are dividing and the telomeres get long and those cells don't enter senescence. But I did not show you that if it's actually already that one cell is in senescence that you can get out of that. Probably not.
Thank you.
Mm-hmm.
Does the same biology apply in the plant kingdom? And I'm wondering about some trees that can live for hundreds of years, and whether it has a similar relation to telomerase or telomeres.
Yes, one of the things that's really very nice to study something so fundamental as chromosome ends is that the mechanisms are conserved across all organisms, that is, all organisms that have linear chromosomes. And so yes, this mechanism does occur in plants. There are a number of plant studies that have been done out there. The telomerase is known, the telomere length is known. But just as in mammalian cells, the telomeres don't determine the maximum lifespan of organisms. That is, the maximum lifespan of human being about 100, 120 years old. That is not determined by telomeres. And we can show that in mice and a number of other organisms. There are probably many different things that determine the maximum lifespan. So similarly in plants, the telomeres aren't going to determine how long that organism is going to live, but the telomeres are carrying out the exact same function. The telomerase is the same, and these mechanisms are conserved.
And another thing, in the complex which includes SERT, is there any supposition that that was introduced evolutionarily by a retrovirus?
I'm not sure what complex you're talking about.
The CERT part of the...
Oh, the TERT.
TERT.
Oh, yes, yes, the telomerase, yes.
Dyslexia.
So telomerase, the telomerase enzyme copies RNA that's part of the enzyme into DNA on the ends of the chromosomes. And so it has hallmarks of what is now found also in reverse transcriptases. But if you actually look back evolutionarily, they're both related to enzymes that copied RNA. And so they diverged later on. So there is some supposition that the telomerase diverged before the reverse transcriptases had diverged, but they are related with the common ancestor.
Good. Thank you for the talk. In your multiple generations of your knockout mice, I didn't see comparative survival curves. And it appears that you can have multiple generations without your telomerase and still have viable progeny that live. Yeah.
You can in both, in mice, for the first 3 generations when the telomeres are shortening. In the absence of telomerase, there is no consequence of telomere shortening as long as your telomeres are long. Once the telomeres get to be shorter, in our case in the 3rd, 4th, 5th generations as they're shortening, the short telomeres themselves then trigger apoptosis or senescence, which causes diseases. So you can live without telomerase as long as your telomeres are long, but then once the telomeres get short, and the same thing is found in human families that don't have enough telomerase. In the early generations, there's no consequence, and it's not until the later generations.
Thanks.
Mm-hmm.
Thank you for your talk. Yesterday, you had mentioned that there is no clinical research or evidence that shows products that are currently out there being marketed to help lengthen telomeres. So can you address whether, I'm assuming that there is research currently going on to try to find, either plant or botanicals or something that will help to lengthen telomeres. Mm-hmm. And I understand that there may not be clinical research studied data, but that there's many, hundreds, thousands of anecdotal data about things like traditional Chinese medicinal herbs that can help to, help bone marrow or make gray hair black again, like Hoshu Wu. So can you address that?
I'm not an expert in any of those sorts of medicines. I certainly know that there is a lot of interest in both sides of looking at telomerase, in finding ways to lengthen telomeres in the setting of bone marrow failure and pulmonary fibrosis, that if we had some mechanism whereby you could lengthen the telomeres and maintain them, that would be really an outstanding treatment from people that are dying from these diseases. So there's a lot of interest in looking at that. And there has also been, on the other side, some interest in looking at telomerase inhibitors as approaches to cancer. But now that we know a little bit more about the normal consequences of having less telomerase, we need to just step back and think a little bit about how long you would want to inhibit telomerase before you may have other effects. So one needs to have a little bit more of a nuanced approach. But these are definitely areas that people are actively trying to understand as we understand the disease, understanding treatments for the disease. My colleagues at Hopkins, see patients every day that are dying from these diseases, and there's no treatment. So we are very interested in pursuing that. Thank you.